Hi everyone, this is about take 5,000 for me. Um, None of my technology seems to be working. So let's see, we'll get the pen set up and hopefully this works. It is leading really ugly lines all over the place. This video may be a little bit longer than the other ones. I'm going to try to keep them around 20-25 minutes so nobody falls asleep. But I wanted to point out some tips that will make your life easier while you study this material. So as you move along, make sure that you are comparing the similarities of the endocrine system and the nervous system, and then contrasting the differences between the endocrine and nervous systems. Also keep in mind that endocrine glands are ductless, unlike exocrine glands, which have ducts and were previously studied in AMP1. So endocrine glands release / secrete into the bloodstream. There isn't a duct that holds any of the hormones until then. All right, so when we cover hormones in the endocrine system, it's going to be really helpful for you as students to keep in mind if you see the word inhibiting or releasing in the name of the hormone, that hormone is from the hypothalamus, okay? Also keep in mind those hormones are going to come from the hypothalamus and regulate the anterior pituitary, okay? So the hypothalamus regulates the anterior pituitary. That's the one closest to your nose. It's going to be, if you're in an anatomical position, it's going to be in front of, hence anterior. So the other name for the anterior pituitary, which I don't frequently use, but others may, is the adenohypophysis, which is PHYSIS, adenohypophysis. I'm sure whenever I get to the closed captionings, that's going to be a lot of fun to fix. So I want to run you guys through a quick example. When the hypothalamus releases a releasing hormone or an inhibiting hormone, it's going to go and act on the pituitary to trigger secretions from the pituitary gland into to direct them to the target organs. So, for example, TRH, thyroid releasing hormone, from the hypothalamus acts on the anterior pituitary, which then secretes TSH, thyroid stimulating hormone, to the target organ, the thyroid. Now, don't confuse the use of inhibiting or releasing and stimulating. So if it's got an inhibiting or releasing, again, it's from the hypothalamus. So the hypothalamus, in this example, is releasing thyroid releasing hormone, okay? That goes to your anterior pituitary, which then secretes your thyroid stimulating hormone to target the thyroid. That then releases thyroid hormone, which we'll also see is thyroxine 3 and 4 later. But keep in mind that the positive and negative feedback mechanisms of thyroid hormone. So to continue this example further, the thyroid hormone itself will stimulate most of our metabolic cells. Okay, so it's going to stimulate those. The same hormone inhibits the hypothalamus from releasing more thyroid releasing hormone. So while that thyroid hormone, or thyroxine, is stimulating metabolic cells, it's also inhibiting the hypothalamus from releasing more. So it's got a positive feedback towards metabolic cells and a negative feedback towards the hypothalamus. So the tips and tricks I wanted to give you guys. I'm really hoping this pen tool is going to cooperate with me. I think I picked it. Okay. So next slide. I'm not going to read these to you. There are 10 of the learning outcomes. Okay. Keep these in mind, even as I make other videos for this chapter. These are what you as students want to master and move on. Make sure you clearly understand so that you can get them correct on the test. So when you're like, what's she going to test us on? It's coming from these learning outcomes. Okay. So keep these in mind. All right. So the endocrine system. There's about 30 different chemical messenger. Chemical messenger. Taking this guy off. Where's my? All right. There's about 30 different hormones, right? Which I also call chemical messengers, hence that. So let's see if I can get this on highlighting mode since the pen doesn't want to work. So there's about 30 of them. Do you need to know all of them? Nah, no, you're probably going to need to know what their abbreviations are and what their mechanisms of actions are. So what do they do? That's what I mean by mechanism of action. So keep those in mind. We're going to go through them all as we progress through the chapter and these recordings. And keep in mind, listening to these recordings once and not reading the book or not doing your homework is not going to help. So there's a list of engaging activities and how you guys meet your goals for learning that if it's not already available on Canvas, I will make available. Now let's see if we can get that silly little pen to work. All right. Or we'll just leave it on a highlighter for now. All right. So direct communication, you guys have probably seen this. Oh, dread. Nope. Back. You guys have probably, why is it not doing this? Highlighter. You've probably seen direct communication. We talked about gap junctions with the nervous system in AMP1. So these are cell to cell across the gap, right? And is also between two cells of the same type. So nerve cell to nerve cell, right? They're very specialized and they're pretty rare. So the other ones we're going to look at that are important are paracrine, autocrine, and specifically in this chapter, endocrine. So paracrine are signals that go back and forth from cell to cell in a single tissue, right? So a single tissue, it could be adipose tissue, muscular tissue, glial support tissue, could be a number of different things. So autocrine, these are remember auto is self-working, that stupid line. Okay. And so these cells that secrete them also get impacted. And there are a bunch of chemicals that are involved in autocrine, and we'll go over them. For example, prostaglandins. We're going to see these when we get to the digestive system. We're going to see how they control the the smooth muscles that are run without us actually thinking about them, okay? So they control the smooth muscle contractions in our digestive tract, and they move the food along as we break it down and start to absorb nutrients. And there's an entire chapter on digestion and an entire chapter on nutrition. So we'll get there when we get there. When we focus on endocrine communication, this bad boy, I can't with that stupid line. All right, we're talking about hormones specifically. And keep in mind, hormones are not like pheromones. Hormones are blood bound. They are found in the bloodstream and they alter a bunch of metabolic activities and impact many, many organs. So you've got target cells. These guys have the receptors they need to read. And here's where I would normally draw a little image for you, but it just seems to lead me to getting frustrated because they don't seem to want to. This tool does not seem to want to let me draw things without drawing lines all over the place. So if we're talking about a cell or a type of diabetes, for example, and here's the cell and here's a receptor, not to scale. So if you have the normal, not mutated version of an insulin receptor, which is this guy here, insulin, that stupid line, is going to fit into this receptor here, okay? If, however, you have a mutated version that we see, that receptor shouldn't be there, but we'll just make it a receptor. This circle, insulin, for my little purposes of diagramming, wouldn't fit into this little triangle area, this little cornered end. It wouldn't fit, which is what we see with different types of diabetes. So keep in mind that for target cells to work, their receptors can't be mutated. And mutations can be genetic, they can be environmental. We're not going to go into all of that, but the receptors have to be able to read the hormonal message that your body is making. So if my insulin is this shape, my receptor is this shape, I'm good. If my insulin is this typical shape up here, and then this is my receptor, whether it was a genetic mutation or an environmental mutation, it's not going to fit. And that's where people run into issues with diabetes, specifically variants of type 2 diabetes. All right, so we have hormones. Generally, they have a pejorative or a negative connotation with them. Somebody's hormonal, blah, blah, blah. We're going to avoid those connotations and keep in mind that hormones are needed for all kinds of changes in our body. The quantities are going to be important, as well as, like we've mentioned, the receptors on the target cells, and that their activities are a bit enzymatic. Okay, they are structural proteins in target cells. So, yeah, we'll get to that. Okay, so they can and will alter metabolic activity, right? So, like I mentioned, thyroid hormone stimulates our metabolic cells, right? But then it upregulates or has a positive feedback on our metabolic activities. Thyroid hormone, right? TH. However, it has a negative impact on the hypothalamus, which is part of your brain. I hope you guys know that. All right. So again, they can affect long-term processes like growth and development. So a lot of sports athletes, or not a lot, but there are athletes out there that are using human growth hormone, right? And they're using it illegally. And I'll talk about it when we get there, but it actually doesn't help them as much as they think it does, but and it leads to things like cancer and other unfortunate things. But it was used in the 90s for for children who weren't growing fast enough, and we didn't understand the consequences back then. Okay, so here again is where you're going to want to compare the synaptic communication we see in our nervous system with the messages and the communication we see in our endocrine system. And both systems are going to be, why is it not showing? Oh, freaking seriously. Okay. Both systems are going to be involved in crisis management for an organism such as ourselves. Right. So these are the similarities, right? And again, you want to make sure that you understand if homeostasis breaks down, that organism is facing, if not a very difficult recovery, possibly death, right? So you have to have coordination between our structures and our functions and how to regulate them, right? Most often, endocrine and nervous system are going to be regulated by negative feedback. That slows things down. It shuts them off, okay? That's the majority of how we regulate our body. It's regularly how we do it. Another one that we'll hear about, and hopefully you've already heard about, is positive feedback, okay? And this is less common, but includes hormones such as platelet-derived growth factor and human chorionic gonadotropin. And that's a G, and that's a C, and that's an H, but that line is really annoying. All right, so these would be positive ones. So these are examples of positive ones. They're less common. I freaking hate this pen. Than negative feedback. All right, so like a really simplistic overview, we're looking at the hypothalamus, and they're only talking about antidiuretic hormone here and oxytocin as regulatory hormones. But like I mentioned earlier, if it's got the word inhibiting, or releasing in the name of the hormone, it's coming from the hypothalamus. Okay, it's going to go in then and act on specifically the anterior or adeno hypophysis, the anterior lobe for your pituitary gland. So the one that's towards the front here, right? Your posterior lobe is, it's not really diagrammed well here, but it's really small. Anyways, they're showing you very limited numbers of what's going on. So like I mentioned earlier, so if thyroid regulating hormone is released by the hypothalamus and goes and impacts and signals the anterior lobe. The anterior lobe is then going to secrete and release thyroid stimulating factor, which goes and impacts the thyroid. Okay. So I know that there's a lot of complex diagrams in this book. You've got me. Crash Course is a really good website too, if you can handle how fast he speaks. Some of my students also really like openstacks.org and Khan Academy. And Khan Academy is KHAN Academy.org. Both openstacks.org and khanacademy.org are free resources that you can sign up for. And when my students get lost by the book or something like that, they'll go to one of these websites and figure it out. If they still can't figure it out, please contact me. All right, so the pineal gland is responsible for, well, they're just saying melatonin, but melatonin helps us sleep. And to get to melatonin, you actually have to consume carbohydrates and serotonin as well, actually. But you guys will see that in a little bit. The parathyroid glands, I'd like to point out, they're pointing to the anterior portion right here. The parathyroid glands are actually posterior on your thyroid. So they are on the back end of the thyroid gland. Just to keep that clear, guys. All right, so here's a little bit more detail. So thyroid gland is going to release thyroxine, which is T4, and triiodide thyrronine, which is T3. And you will most commonly hear me say T3 and T4. When we get to the nutrition section, you're going to want to know, we'll get there, but I'll remind you, what are these guys derived from? So when we get there. So that's just the final, the breaking down the glands, right? And the pancreas, if I haven't already said so, the pancreas is both endocrine and exocrine. So you would have covered that a little bit, hopefully. If not, we're going to see its roles as we move through the class. Okay, so here's the key. So TSH, thyroid stimulating hormone, and a whole bunch of them. I'm not going to name them all, but there have been some changes. Like this one is different from this been changed in the last 5 years. Occasionally I trip up and call it something else, but I will do my best not to do that to you. And then you'll see this is why we start this course out with endocrine, because it's going to impact basically every other system we go over this semester. So if you want to read ahead and see where their functions, the secondary functions are, go ahead and pop in to any of these chapters and look and see how important it is, right? Okay, so classes of hormones. We have three basic classes. Sorry, amino acids. You don't need to know the whole structure. It would be nice to know if you knew that there was a carboxy group on one end. So we'll just do an HOOC here. Oh, that line wasn't smooth there. An alpha carbon, uh, an amino group, which is where I get this name from. A little tiny hydrogen hiding down here, and then a big old R group. And this is where we studied 20 different ones. There's actually 27, but you don't need to know all 27. You don't need to memorize all 20 right now. Right. So we see amino acid derivatives. These are going to be small. And do you need to know the structure? No, you don't need to memorize the whole darn thing right now. I'm just going to get an eraser out, but you don't need to memorize the whole thing. Wow, this is really not letting me work it. You don't need to memorize all this right now, okay? We will get to what you guys want to understand. What you want to know now is that the amino acid derivatives are really small. They're going to be very small. Whereas you have peptide hormones, and they're going to be 10 to 20 amino acids long. And when I abbreviate amino acids, I use AA. So 10 to 20 amino acids long. I really wish I had a better tool, guys. I'm so sorry this is going on. Okay, so the peptide hormones are between 10 and 20. The amino acids are typically less, oh my gosh, less than 9 long, and they can be even 2 or 1. The lipid derivatives we're going to get based on our lipids we pull out of our diets. So there are some diets that are new, fad, all the rage or whatever, and they'll either deprive you of lipids, or they'll deprive you of carbohydrates. And that means you are not able to make certain hormones, which means you are going to find an imbalance and you're going to suffer. It could be mood swings, it could be lack of sleep, it could be many, many things. So make sure you speak with a registered dietitian before you pick up a new in style diet. All right. So again, the amino acid derivative hormones, they're also called biogenic amines. They're really small, and we get them from our amino acids, right? Tyrosine is one, right? And then tryptophan is another. So without tyrosine, we wouldn't be able to get our thyroid hormones. We wouldn't be able to get our catecholamines. Sorry. And to remember catecholamines, I use Ned. So like Ned Flanders from The Simpsons, but a norepinephrine, epinephrine, and dopamine. So that's one of the ways I remembered catecholamines. So I said Ned was nice to cats or something like that. The tryptophan one, this is where we get our serotonin and our melatonin from, like I was mentioning earlier. So serotonin, this one will prevent depression. It allows, one of the ways we treat acute or chronic depression is by using serotonin reuptake inhibitors. I forget what the first S stands for, but serotonin reuptake inhibitors, and that'll get brain neurons that either are not making enough serotonin or not, they're taking back up too regularly. It'll slow down the uptake, and from that, the reuptake, and that the secreting axon, and allow the serotonin to be reabsorbed more quickly. So again, if you don't have enough tyrosine or tryptophan in your diet, you're going to suffer from different things. Melatonin helps us sleep. So this one is also, again, to make melatonin and serotonin, you need different things in your diet. And yeah, I'm throwing a lot at you guys, but I'm just trying to help you guys cover all your bases so you understand and you have a good background of what's going on. So melatonin, you can get over the counter, which is abbreviated OTC. OTC, over the counter, in a gummy form or in a tablet form. If you're having problems sleeping, don't take it when you're during the day, but melatonin will help you sleep. There is a very common misconception that you can only get tryptophan from turkey. That is absolutely incorrect. Tryptophan is one of the, oh my gosh, I hate this tool, 20 essential amino acids. Okay. And even vegetarians who don't eat any meat or any turkey, because turkey is meat, still have tryptophan in their diet and they're doing just fine. All right, peptide hormones, the second type. All right, these guys again are short, small peptides. So they're going to be 10 to 20 amino acids long. Prohormones just means that it has not yet been activated or properly converted so that they can be active. Glycoproteins include sugars, right? And a sugar protein part. So they have a glyco portion and a protein part. The proteins on these are actually usually more than 200 amino acids. Not that you need to memorize it, but here are some examples. So, thyroid stimulating hormone, luteinizing hormone, and follicular stimulating hormone. And the best way for you guys to do the hormones and memorize and remember what they come from is use flashcards. So that's what I would recommend there. The short chain polypeptides, antidiuretic hormone, and oxytocin. You don't need to memorize the number of amino acids, guys. But I would say definitely know this one's going to prevent you from losing water. This one helps bonding. Small proteins are going to be, don't confuse these with the amino acid ones, but or amino acid derivatives. These are small amino acids, but small proteins are less than 200 amino acids long, and they include insulin, human growth hormone, and prolactin. This has to do with sugar uptake when you've eaten. This has to do with proper growth, and this has to do with milk dropping into the mammary glands when women are lactating after childbirth. So all of these, again, remember if it has releasing or stimulating in the name, it's from your hypothalamus, okay? But these peptide hormones are from the hypothalamus, the heart, the thymus, the digestive tract, your pancreas, and then I always put little p for posterior, and then pituitary. So gland. If you guys have a great, cool little fun way that's not offensive to remember this, go ahead and we'll put it up in the chat or the posts. Okay. All right, lipid derivatives. The last one we're going to look at. So icosanoids. I'm making that so much more complicated than it needs to be. This is pronounced ico. This is really slow. Synoids. And it comes from a fatty acid. So again, you need to have fatty acids in your diet. But it includes the paracrines we talked about. That are and the paracrines act on their own cells. Let me go back here and make sure I've got the same wording as the PowerPoint. Apologies, guys. So it's cell to cell, not same cell. Apologies. Autocrine is same cell. Oop, this is really ugly. So icosanoids. All right. So, anyways, paracrines is they're going to be involved in things like blood clotting, which is very important. But there's other cellular activity, and it's going to be enzymatically led. Leukotrienes, which we'll talk about when we get into blood. These guys are going to have a secondary role for hormones. And then prostaglandins, which you'll see me abbreviate as PG. That's a fracking G right there, or sometimes PGS. And this is going to help with cellular activities at a local level. So we're going to talk about thromboxans. Not really going to cover prostacyclins very much, but in your steroid hormones, there's I don't know how to say this nicely, a prude American way of thinking that only males have androgens, and only females have estrogen and progesterone. That's not correct at all. In fact, to get to testosterone, you need to make a form of estrogen first. It's called estradiol. Do you need to know estradiol? No. But do you need to know that testosterone is derived from estrogen? Yeah. You can't grow, cell growth cannot occur without estrogen. Okay. And musculature will not expand. Skeletal density will not improve if you don't have testosterone. So, both biological sexes in the human species have testosterone and estrogen. Okay. Corticosteroids, these are the ones that dump when you're going through something stressful, which is technically distress. And then we'll see calcitriol from the kidneys later towards the end of the material we cover in this class. But keep in mind that the lipid derivatives are going to be, bound to specific transport proteins in the plasma, and they remain in circulation longer than peptide hormones, which bind. All right, so to transport and inactivate hormones, you're going to have to consider that there are free hormones and then there are bound hormones. And then we have special carrier proteins for them. So free hormones by themselves remain functional for a shorter amount of time, and they will diffuse out of the bloodstream and bind to receptors on their target cells. Okay, once they're absorbed, they get broken down by the liver and kidneys, or I should say, or kidneys, and they also get broken down in the blood or interstitial fluids between organs. Okay, so you've got thyroid and steroid hormones. These guys will remain functional far longer. About 99% become attached to these special transport proteins in the blood. They're plasma components, but they'll get attached there, and then these will actually find, hopefully, a state of equilibrium between what is free and what is bound. What is bound and what is free. And then again, we're going to use our bloodstream as a reserve for bound hormones. So the plasma components can let go of them if they're needed. Right. Hormone activity, I wanted to point out that there's 3 levels of hormone action. You can alter the DNA level or genetic activity, the mRNA protein level, and then cell level by basically changing the permeability of the cellular membrane. So keep that in mind. The mechanisms for them to bind are going to be, I'm going to go all the way into the nucleus and impact DNA. I'm going to hang out in the cytosol and impact protein synthesis. Or I'm just going to hit the edge of the cell membrane, right? And I'm going to impact that. So keep that in mind. It's always nice to draw things out and have a good understanding. And if you can't explain it in words, start with a drawing and then add words to it. And if you work the other way, start with words and then try to make a drawing. And if you can explain it to us, a classmate or back to me, then you know it. All right, hormone receptors. So it's going to be a protein molecule um, that binds specific or particular molecules very strongly, right? And then different tissues have different combinations of receptors. So, for example, if you're looking at the adrenal epinephrine or adrenaline receptors in males versus females, females have more receptors for more adrenaline receptors. So more epinephrine receptors. Males have fewer. So an adrenaline dump or an epinephrine dump in a male, well, they don't have as long-term of a response. Whereas, because for some reason on the evolutionary scale, females have more receptors, their adrenaline response or epinephrine response is going to be prolonged. So, should there be a difference in the prescriptions of an EpiPen? The sorry, EpiPen prescriptions based on sex? Probably, but you'd have to test the individual for the number of receptors. So before I digress any further on that, um, if there are some specific receptors that are present or absent, that is definitely going to have an impact on determining the sensitivity of a cell. So if you can dump all the hormones you want on a specific cell type that's in a growing petri dish, if they don't have the receptors, they're never going to respond. Whereas if they have the receptors, they could have maybe a few and be not very sensitive, or they could have a billion and be highly sensitive. So keep those in mind. So downregulation, you will commonly hear me use this as negative feedback. Depending on if you're in bio 1 or 2 right now, your instructor may use it differently. So downregulation, we're going to slow things down. We want to decrease what's going on. So you got a hormone that's present, it triggers a decrease in the receptors. So we're making this a smaller impact. When the levels get high, the cells become less sensitive. Okay. So the higher the hormone is, the less sensitive it is, the less likely it is to respond. Again, down regulation or negative feedback. Upregulation, when you have a hormone that's absent, it's going to trigger an increase in the number of hormone receptors because your cells are like, wait, well, why isn't this hormone present? What's going on? I need some receptors going here. Let's go. What's going on? All right, so keep that in mind. So you'll have an increase in the number of hormone receptors when a level gets really low. So the levels of hormones become really low. The cells become more sensitive to it. Okay. All right, catecholamines. Again, if you remembered NED, you already know that we're talking about norepinephrine, epinephrine, and dopamine. Right? These guys are not fat soluble. They're not lipid soluble. So that means they cannot get through the plasma membrane of the cell. Instead, they're going to interact with the surface of the plasma membrane on all these extracellular receptors that I've been trying to draw on unsuccessfully. Oh, it worked. Okay. Whereas steroid and thyroid hormones, these guys are lipid or fat soluble. They do, in fact, cross the cell membrane, and they're going to interact with receptors that are inside the cell, right? So intracellular receptors. So if I go back to these 3 levels, would be a 3 right there. Stupid tool. If it's intracellular, right, it's again membrane permeability gets through it and then goes in the cell. And there are different levels of interaction inside the cell, but that's where I'm going with that. Okay, so I want to cover this one before I pick up and go into the other ones. So we have first messengers and second messengers. The first messengers are going to be the ones that actually bind the outside receptor outside of the cell, and they release or they promote the release of the second messenger in the cell. So you have usually there's an intermediary molecule, a molecule in between that appears because the hormone interacted with the receptor. It bound it and it said, "Hey, I've got an interaction going on. Let's get stuff going on. Let's get things started." All right. So it can be an activator, it can be an inhibitor, or it can be a cofactor. So we'll get into that a little bit, those differences later. Not much later, but soon. And then they're going to change again the metabolic reaction rates, which is how I mentioned the ones I mentioned. So examples you want to know for sure are cyclic AMP, so cyclic adenosine monophosphate, and cyclic guanosine monophosphate, as well as calcium. So we're going to see these. And you're going to want to be familiar with this one, and this one. I try to do that for you guys. So you got to know what you got to know. All right, process of amplification. So building things up. The analogy I like down here is when you have a small number of hormones. So say you start out with 3 little hormones up here, right? They bind to a receptor on a cell. Like that's all one binds here, okay? Thousands of messengers then may appear inside the cell, right? And I'm trying to make a difference in size or whatever. But anyways, so if you have one and it caused 100 to pop up and all 3 of these bound, you could see 3,000. So if you've got the snow, apparently it's really windy on my hill right now for snow. You got a snowball. You start it from up here and you let it roll downhill. By the time it gets to the bottom of the hill, it's big. So the other words you'll hear for the amplification are cascade effect. And as it gets bigger, or as the messages go on, you started off with one and end up with hundreds. Start off with one and get really big. You can end up with millions and billions of these second messengers that are amplifying. And I'm going to get to G proteins and pick up here. I hope to see you guys in the next video, if it'll ever let me get out of here. Shenanigans. I think I'm stuck.